Hello, welcome to Chapter 15, Selective Breeding 15.1. Our goal is to describe how mutation and genetic recombination increases genetic variation. The far side says, tough luck, Rusty. 7 years bad luck. Of course, in your case, that works out to 49. I think most of you know what they mean by that. All right, our objectives explain the purpose of selective breeding and also explain how people increase genetic variation. Let's think about it. Where does popcorn come from? Would you be surprised to learn that popcorn is one of the earliest examples of human efforts to select and improve living organisms for our benefit? Corn, as we noted, was domesticated about at least 6,000 years ago by Native Americans living in Mexico. A tiny kernel of popped corn found in a cave in New Mexico is more than 5,000 years old. Objective 1, explain the purpose of selective breeding. So the differences among breeds of dogs are great. So where do these differences come from? Humans use selective breeding to produce animals with certain desired traits. Selective breeding allows only those animals with wanted characteristics to produce to the next generation. For thousands of years, we produced new varieties of cultivated plants and nearly all domestic animals by selectively breeding for particular traits. Native Americans selectively bred tisonite, which is a wild grass native of central Mexico, to produce corn, a far more productive and nutritious plant. Corn is now one of the world's most important crops. There are two common methods of selective breeding: hybridization and inbreeding. Let's look at hybridization first. I'm having a hard time saying that word for some reason. American botanist Luther Burbank developed more than 800 varieties of plants using selective breeding methods. One method Burbank used was hybridization. Got it. Crossing dissimilar individuals to bring together the best of both organisms. Hybrids, the individuals produced by such crosses, are often harder than either of the parents. Many of Burbank's hybrid crosses combine the disease resistance of one plant with the food-producing capacity of another. The result was a new line of plants that had the traits farmers needed to increase food production. July Alberta peaches, for example, as you can see right here, are among Burbank's most successful varieties. Inbreeding. To maintain desirable characteristics in a line of organisms, breeders often use inbreeding, the continued breeding of individuals with similar characteristics. The many breeds of dogs, I should say, are maintained using inbreeding, ensuring that the characteristics that make each breed unique is then preserved. Although inbreeding is useful in preserving certain traits, it can be risky. Most of the members of a breed are genetically similar, which increases the chance that a cross between the two individuals will bring together two recessive alleles for a genetic defect. Our objective is to explain the purpose of selective breeding. So humans use selective breeding, which takes advantage of naturally occurring genetic variation to pass wanted traits on to the next generation of organisms. Objective 2 is explain how people increase genetic variation. All right, so when scientists manipulate the genetic makeup of an organism, they are using biotechnology. Biotechnology is the application of a technological process, invention, or method to living organisms. Selective breeding is one form of biotechnology important in agricultural and medicine, but there are many others. For example, mutations occur spontaneously, but breeders can increase the mutation rate of an organism by using radiation or chemicals. Many mutations are harmful to the organism, but breeders can often produce a few mutants, individuals with mutations, with useful characteristics that are not found in the original population. For example, scientists have developed hundreds of useful mutant bacterial strains by treating bacteria with radiation or chemicals. Certain strains of oil-digesting bacteria are effective for cleaning up oil spills, and scientists are currently working to produce bacteria that can clean up radioactive substances and metal pollution in the environment too. Drugs that prevent the separation of chromosomes during meiosis are very useful in plant breeding. These drugs can produce cells that have many times the normal number of chromosomes. Plants grown from these cells are called polyploid because they have many sets of chromosomes. Polyploidy is usually fatal in animals, but plants are much better at tolerating extra sets of chromosomes. Polyploidy can quickly produce new species of plants that are larger and stronger than their diploid relatives. A number of important crop plants, including bananas, have been produced this way. So objective 2 is explain how people increase genetic variation. So breeders can increase the genetic variation of population by introducing mutations, which are the ultimate source of biological diversity. All right, objectives again was explain the purpose of selective breeding and explain how people increase genetic variation, which support our goal to describe how mutation and genetic recombination increases genetic variation. And back to our original cute kitty pictures. Enjoy!